I'd like to start by thanking everyone for logging in today and watching all of our Equalize presentations. So I'm Rebecca Keate. I'm CEO and co-founder of Cellmen Technologies. At Cellmen Technologies, our mission is to improve the quality of life for patients with chronic, non-healing, and infected wounds. I have previous experience developing products with big companies such as Johnson & Johnson, where I worked in wound care, as well as at Becton Dickinson or BD, where I worked in diabetes care. But in addition to working with larger corporations, I also have worked at a smaller startup called AlloCare Health, where I learned about all the different hats in the fast-paced environment of a startup and really enjoyed the culture there. I founded Cellmen Technologies with my PhD advisor, Dr. Guillermo Amir, who is a professor of biomedical engineering and surgery at Northwestern. Dr. Amir has a track record of successfully developing technologies in his lab that later went on to be commercial products, including one technology that's currently being sold by Stryker. So today I'm going to talk about wound infections, how they continue to occur, yet current solutions are insufficient to address these, particularly because they're so damaging to skin. Our product is based on panthenol citrate, which is a novel molecule, which we're using to build a platform of better advanced skin and wound care products that can both sustain and elevate skin health. So we're currently IP protected through a compositional patent. We've raised $90,000 to date through Northwestern's Query Incubation Lab, and we are also completing in vivo studies in a large porcine wound model. So I want to start by just laying out what this problem is. Surgical site infections specifically are the wounds I'm going to talk about today, and they're a major problem in the US as well as globally. And you may not be familiar with how prevalent surgical site infections actually are, but they occur with up to 5% of all surgeries where an incision is made. And the cost of treating someone's infected surgical site amounts to about $5 billion in supplemental US spending alone. Now, in addition to the spending and the cost of treating surgical site infections, a patient's risk of death can increase by up to 11 times when they experience an infection. So this has a really severe effect on the patient's health. But in addition to that, the hospitals care about these problems because they are directly penalized through Medicare when their surgical site infection numbers get too high. In addition to surgical site infections, any sort of readmission associated with this infection has really severe consequences. And this amounts up to about $250 million per year that hospitals are withheld money from Medicare. So although hospitals are really invested in this problem, the current solutions aren't necessarily addressing it. So there are a couple of ways that hospitals try to stop surgical site infections. The first is through a pre-surgical skin colonization, which is applied to intact skin. An example, chlorhexidine is among the most popular product that's used. However, it can cause severe allergic reactions in some patients. And these reactions can be so severe that in 2017, the FDA actually issued a warning about the consequences from the use of chlorhexidine. Even if chlorhexidine doesn't cause a severe allergic reaction, it still is very painful for the patient. And I pulled this review from drugs.com just to highlight words like burning and irritating. And at the end, the patient is saying there must be an alternative, because overall, the use of this product on pre-surgical intact skin is still really damaging and irritating. Now, there's also an approach to apply prophylactic products post-surgically onto the wound. However, a lot of these products that are used, such as hydrogen peroxide or Dakins or chlorhexidine as well, impede healing and hurt the wound even more. So they're not widely used because surgeons just don't think they're going to help. So because these products are harmful to regeneration, overall, there's this overarching theme that these products that are currently used to prevent infection are really damaging. So we sought to develop a better skin healthy solution based on natural precursors, citric acid and panthenol, which are already widely used in a variety of skincare products, such as lotions, cleansers, and creams, to promote skin health without damaging it. Our technology is a synthetic molecule that's based on panthenol and citric acid called panthenol citrate. And because it's synthetic, we're able to protect it through a compositional patent. It's also highly anti-inflammatory, which further differentiates it from the other products that cause allergic reactions or can irritate the patient. And finally, there's this really interesting property of panthenol citrate, which is its luminescence, and that enables it for monitoring in the case of infection. So we can potentially identify it before it even occurs. Now, because of panthenol citrate's multifunctionality, as well as the diverse needs of wounds, there are a lot of different areas that we could potentially address with our product. So in order to inform our business strategy, we performed in-depth customer discovery, talking to different physicians, nurses, and even patients. And we found that we were able to evaluate each of the different categories we could address against different risk levels. And so with that, we developed a pipeline where we start with prophylaxis, move into eventually potentially making claims about infection treatment. Later, develop infection identification products once the technology is more ready, and then eventually even make claims about improved healing. But for the rest of this presentation, I'm going to be focusing specifically on our products for prophylaxis. And so in order to be a prophylactic product, there are two main functions that a technology needs to achieve. It has to be antimicrobial, and we've demonstrated that against the three major infectious strains, panthenol citrate has antimicrobial activity. It also needs to work for a sustained period of time, and we've shown that over several hours, panthenol citrate is able to sequester bacterial growth in a highly pro-growth environment. In this case, broth was used. But in addition to being antimicrobial and sustaining these effects, we thought about the economics of this. And so chlorhexidine is currently widely more used than povidone iodine, which is the second leading product. And that's because every surgical site infection that you prevent saves the hospital $5,500. So even though chlorhexidine is a higher product cost, since it performs better, that saves the hospital more money. And so we believe that we have estimated we can hit the exact same cost as chlorhexidine, but that leaves us with the question is, how are we going to be better? And the answer is inflammation. Several studies have demonstrated that high levels of inflammation lead to a higher risk for surgical site infection. And because panthenol citrate is anti-inflammatory, we've shown in a diabetic mouse model that using this molecule increases anti-inflammatory molecules, cytokines, while decreasing pro-inflammatory cytokines. So overall, we're preparing the skin with a better reagent that's going to reduce inflammation and reduce surgical site infections. And so these are the main products that we're going to be competing with. Overall, most of them are not approved for both pre and post-surgical use because they either are oxidized very quickly and don't have sustained effects, or they're not safe for promoting healing. In addition to all of the ways I've already mentioned that our technology will exceed those capabilities, we also identified a gap in the pediatric markets. So while these technologies are currently used on pediatric skin, we widely observe a number of rashes or inflammatory responses. But we think this is an area where we can uniquely address the needs of pediatric patients that are currently unaddressed by these technologies. So overall, we're currently working on collecting more data in an infected wound model that was recently developed in our lab and evaluating panthenol citrate's activity against resistant bacteria. We're seeking a $4 million seed fund to get us all the way to commercial launch, where we believe we can start collecting revenue. And at that point, we're also going to be continuing to develop out our luminescent products, which will continue to add value to our company. With the $4 million seed round, the formulation prototype is the most pressing milestone that we have to achieve, followed by 510(k) approval, And then we plan on completing a clinical study for marketing purposes. So just to summarize what I've spoken about today, existing surgical wound infection solutions fail to offer skin-healthy solutions. Panthenol citrate can prevent harmful bacterial colonization. And I also want to reinforce that this is a platform. So I just showed a picture here of its luminescence, and that's going to be something that we continue to develop to add value. Our major milestones include a formulation prototype, seeking 510(k) approval, and completing a clinical study for marketing purposes. So with that, thank you again for listening to my talk today, and I'll take any questions.